Everyone, I'm Kira Curtin, Senior Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Revolutionizing Personalized Medicine Through Metagenomic Sequencing Strategies, and is sponsored by Complete Genomics. Our speaker today is Maike Seifert, head of the Lab Core Unit at the Center for Translational Microbiome Research at the Karolinska Institute. You may type in a question anytime during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. And if you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. And with that, I'll hand it over to Maike. Maike?
Hi, good afternoon or morning, everyone. I'm Maike. As Kira already mentioned, I'm the head of the Lab Core Unit at the Center for Translational Microbiome Research at Karolinska Institute in Stockholm, Sweden. And I'm also the CEO of Biogen, which is a newly founded startup company that is providing sequencing service for metagenome sequencing primarily. Today, I'm going to talk to you about how we think of revolutionizing metagenome sequencing strategies for precision healthcare and how we are doing this. But I'm going to start with, with talking a bit about who we are and what we're doing. So at the Center for Translational Microbiome Research, we aim to better understand the contribution of the microbiome to different health and disease states, with the aim to open up opportunities for development of novel therapies in the areas of gastroenterology, reproductive health, and cancer. We have been founded in 2016 as a collaboration between Ferring Pharmaceuticals and Karolinska Institute. And since then, we have established a broad technological, biological, clinical, and epidemiological platform to study complex microbiological communities in defined human materials. And today, the platform includes a biobank of more than 200,000 human samples with clinical metadata and more than actually 2,000 bacterial isolates with phenotypic and genotypic screening pipelines for consortia selection and biopharmaceutical drug development. I still need to get used to this. The CTMR research areas are, as I mentioned, gastroenterology, cancer, and women's health. But in order to understand how the microbiome influences health in disease states, you need to understand how a normal microbiome looks like and define a normal and healthy state in relation to different disease states or pathophysiological states. And in order to figure this out and to define a normal state, you need to have large-scale microbiome studies. The microbiome is influenced by a lot of different factors, and we all know today that each individual microbiome is unique, and therefore large population scale studies are needed to define certain biomarkers that have influences on different pathophysiological states. And this is how you could go about if you would want to look into this, or how we chose to go about. So since the Karolinska Institute is very closely related to Karolinska Sjukhuset, the Karolinska Hospital, we have a very broad clinical network that we are utilizing and who is utilizing us to set up those big population-based studies, to do sample collection for the studies to investigate the role of the microbiome. From then on, you could, or we have, go on to develop in-house technologies and assays, phenotypical and genotypical screening pipelines, to establish analysis pipelines, to find biomolecule targets and lead candidates that are involved in different pathophysiological states or health states. And next to this, we also focus on developing different culture medias for large-scale bioreactor processing to, in the end, then be able to culture, characterize strains, and semi-defined consortia and defined assemblies that have the potential to be adapted for personalized therapies and can then further be used for genomic mining for functional therapeutic opportunities. The human microbiome is further changing our perspective on health and disease, but we have yet to see that we have recently started to see, but we hope to further see how it's changing our therapeutical practices. And we believe that in the future, microbiome components can be used as biomarkers for early detection of different pathophysiological states, that the microbiome components can have a direct impact on human physiology and pathophysiology, that there are opportunities for the modulation of the microbiome, and that those further can be used for precision medicine in the future. This is our general toolbox. So we're working with metagenomics and metatranscriptomics, where we're using different assays for exploratory research. We have a handful of metabolomics pipelines, and as I already mentioned, we have a quite substantial culturomics unit that is working with phenotypical screening pipelines and the development of culturing media and the establishment of consortia. But when we set up the center, and here in this talk, we're going to focus primarily on the genetics toolbox. So when we found the center, we in 2016, we worked mostly with 16S sequencing, but together with Ferring Pharmaceuticals, we decided that in order, as I explained previously, in order to define a normal state for the microbiome and in order to be able to target certain biomarkers, we would need those huge population-based studies. So there was a need for high-throughput sequencing. And so we went out and compared different high-throughput sequencing strategies, and in the end, decided to build our metagenome sequencing platform on the DNB-seq technology. And the DNB-seq technology delivers certain aspects to the platform that we thought were important as a platform head. I'm mostly looking for to get the most for my buck. No, so you want to do sequencing as cheap as possible, as fast as possible, with the biggest, highest quality as you can. And we can see this, or we are seeing this, and we decided to go with this strategy because of certain aspects of it. And I'm going to talk a bit why those are and how this comes. So the library preparation is quite normal library preparation, as you would do for most pipelines. But then from your library, your adapter-linked library, you go into denaturing your double-stranded DNA fragments and ligate them into single-stranded circles that are extremely stable, so you can preserve them for a relatively long time which makes planning a lot easier. And then when you have a single-stranded circular library, the DNB forming process that is unique to this process is facilitated by a rolling circular amplification that is on the original single strand. And on the next slide, we will go a bit in more detail into how this process looks. But then this DNB, which is a cluster of the original molecule, is loaded onto a patterned flow cell. And this is a bit of a busy slide. So you can see the process that you can see on the slide before. So you have the rolling circle amplification, which is also depicted in the video. So you have the polymerase running in circles on the original strand, which is different from other cluster formation processes where you have an exponential amplification of the original strand, but with an exponential amplification of the original strand, you can easily exponentially build in errors. And since you're running on the same original strand in this form of cluster formation, you do not see clonal errors or index hopping, and you have a very, very low duplication rate, which in the end leads, if you have a lower duplication rate, the final output in the total amount of reads that comes from your flow cell, if that is comprised of more unique molecules, you get more information for the amount you paid. So we see lower error rates and a higher unique count using this strategy compared to other strategies, which was one of the factors that made us choose this platform. Another thing that we thought was important as a platform was that we have a big variability in the platforms that we can choose from, while not needing to change our library preparation process too much. So we wanted to have various platforms that have a different output, so we could facilitate, run projects as fast as possible, but also be able to deliver our results as fast as possible if the throughput is lower than average or higher than average. And there we have been exposed to a broad portfolio that Complete Genomics is having on the market, which goes from the lowest output sequencer, the E25, that gives you roughly 7.5 gigabases, to the highest output platform, the T20x2. And today we have the E25, the G400, the G99, and the T7 in our lab. So we have a huge variability for different kinds of projects, from isolate sequencing to amplicon sequencing to metagenome sequencing, and can run all of those in the same speed. So even if we have isolates coming in that normally don't need as many reads, we can sequence them as fast, and they don't need to wait for a bigger sequencing project to be able to be run immediately. In the times before, we only had bigger sequencing platforms at our disposal, and then we always needed to wait to run an isolate to have, when we had enough space on metagenome sequencing flow cell, which is quite time consuming for our collaborators, and quite annoying in the lab to handle, quite frank. So this is a bit on how we chose to go for the platform to begin with. So we saw this need for high throughput sequencing. We did our evaluations of all the different platforms, and then we chose one. But there was another thing that happened in January or shortly after January 2020, when we started to set up the first sequencers and ran our first full metagenomes. COVID hit, and we needed to close down our lab, like most other labs. But because of our new collaboration with Complete Genomics, we were very fortunate to get their support during COVID. So together with their support and Karolinska Institute, the support from Karolinska Institute and local foundation, we were able to set up a COVID diagnostic lab and a COVID strain surveillance lab with the same infrastructure that we set up originally for metagenome sequencing, which is another part of flexibility that we gained in our lab that I couldn't foresee that we needed at that point, but that I was very grateful for. So with the same infrastructure, both in liquid handling and on the sequencing platform side, we completely remodeled our lab and made it into a diagnostic lab. And this is here on the slide, you can see a bit how we set this up. So we have 4 STP-7000, which is a sample transfer robot. So what the robot does in reality is it takes tubes of various sizes and forms. It uncaps them, it takes off the screws automatically, and then it pipettes them into a deep well plate and puts them back together, and gives and reads the barcodes and makes sample sheets for them. Those we have 4 of. Then we have a smaller SP100 that is mostly used for DNB making and smaller library preparations. We have 13 SP960s, which are quite sturdy liquid transfer handlers. So they do liquid transfer with a 96 well channel head, which we use for RNA and DNA extraction, as well as for library preparation, for isolates, and for metagenomes of various kinds. 4 DNB-seq 400 sequencers, so those are in the mid-output range, producing 1,600 million reads per flow cell, and 1 DNB-seq T7. This is a higher output sequencing platform that is producing between 5 and 6 billion reads per flow cell on 4 individual flow cells that can be run independently from another. So this is the setup we were planning to use for metagenomes, and then could very quickly transfer into an amplicon sequencing pipeline for a completely different purpose. So we run both diagnostics and SARS-CoV-2 whole genome surveillance on this. And there you would do the amplicon with a tiled amplicon approach in a 2-step PCR, where you amplify the SARS-CoV genome, and then you add the needed adapters and indexes, and then you have a fully formed library that you can then use to sequence the amplicon libraries, even though they're in the end, because of the tiled multiplex approach with 260-ish primers, give you a complete representation of the SARS-CoV genome. And as you can see here, the same pipeline, the same platform. We have done in 2022 retaken, I don't think that's how you actually say it, but restructured in order to do microbiome or metagenome sequencing again. And there I also quickly want to talk you through how we do this as a center. So in metagenome sequencing, you want to include as little bias as possible, because there is already so much noise in the data. You want to keep the noise as low as possible in order to identify the biomarkers, of which we still up to date are not totally clear on if there would be more low abundant or high abundant players in the community. So looking for low abundant players, you want to reduce the noise obviously as little as possible, and therefore we like to take as much control over the study as possible and include as many controls as possible. So in an ideal scenario, we would work together with our clinical collaborators on defining the study and the study material. We then would prepare the study material and then send it to our collaborators that then send it out to the patients or use it on the patients depending on the study. And then we would get the the sampling material back to us, because we have been involved from the beginning. We also have control over the barcoding. So you can see we have our own freezer storage system with an internal laboratory management system for the freezers. So we at all time know where every sample in our freezers are in the biobank. And then we have a pre-labeled sample tube system that is labeled on the side and the bottom of the tube, as well as on the plate that is holding the tubes, that is then used to track the samples at every step. Once a study is concluded, we decide together with the PI how to do the analysis of the sample, and then we make sample lists to include all the samples that we decided to work on with the appropriate controls and our internal controls. So when we design a study with the collaborators together, we normally include controls from the environment, even of the sample taking process. Then we have environmental controls in our lab, and we also have in-sample controls to be able to characterize the contamination if there is, and the efficacy of the process in each sample, as well as positive and negative controls for the process that are in every plate for both the extraction and the sequencing side. Starting with the extraction, we would then perform a fully automated, semi-fully automated extraction on the liquid handlers. To date, we, as I said, we use a variety of the SP960 for this, and another liquid handling system that is coming from Tecan. And after the fully automated process, the laboratory process that includes extraction and library preparation, we would then decide a feasible sequencing platform, sequence them, and then they automatically uploaded into our storage servers and processed automatically or pre-processed automatically, and then distributed to the bioinformatics team, either in-house or external bioinformaticians, depending on the project. And during 2022-23, we ran both of those pipelines actually in parallel in the same lab on the same machines. And it's something, as I said before, that I couldn't foresee as a lab manager at this point, but that I'm today extremely grateful for, and that I wouldn't do any. So if I would set up a platform again, it would be something that is important for me because it's quite an investment. You want your machines to not be able to only fulfill one single task because you never know when the world suddenly is changing. In order to do those processes and to do the best metagenome sequencing we did a lot of development. In one of the earlier slides, I talked about technological development for the pipeline. And part of the technological development is to set up standardized procedures for sample handling that give you the best possible outcome in terms of you want to realistically depict your metagenome. You want to see all the players that you have in your community. We have up to date a standardized sample pipeline for fecal samples, vaginal samples, biopsies, saliva samples, gum cavities, and skin swabs. And when we started to develop this pipeline, we were a bit unsure on how to go about with this. So we looked into different positive controls and different defined consortia, and saw how well all the different extraction processes are actually performing, and how well our in-house method is performing compared to what happens if you send this to different vendors and let them extract, and how we compare to not our competition, because we're a state organization, but to other players in the field that have done this for a very long time. And I was quite shocked to see that If you look on the taxonomic plots, on the Kroner plots on the right-hand side, you can see on the top the standard. This is how the standard theoretically should look like. In colors diverted into gram-positive and gram-negative bacteria because they have a different membrane structure, different cell wall structure, and that makes them more difficult or more easy to break. And ideally, you want to break them open all evenly. So you don't want to go about it too harshly, because if you go about it too harshly, you will open up even those very hard to break cells, but then you will also destroy the DNA that you have already managed to expose in the beginning of the process. But you don't want to go about it too softly because you want to get all of the material. So you want to get all of the cells that you have. And for vaginal samples, the microbiome also includes different fungi. So those are next level when it comes to getting out getting out the genetic material. And then we sent those samples out, and we saw that from 2 different vendors, when you look at the distribution of the gram-positive and gram-negative bacteria, we have a very skewed distribution that is not really realistically depicting how the community looks in reality. So we put a lot of effort into optimizing our processes to be able to depict this community as good as we can. And after we were able to do this, and we're able to do this without damaging the DNA too drastically, we went into sample types that had lower biological material. So when you get a fecal sample or a vaginal swab, you normally get quite a lot of biological material. When you get a skin swab, which is someone rubbing a cotton pad on a bit of wet skin, you get very little biological material and even less bacteria. So we optimized our process further to be able to prepare a library without actually having minimum input requirements. So we can make a library out of only a very minute amount of cells, equally good as we can make a library out of a lot of biological material. As I said a bit earlier, today we also use spike-in controls for both extraction and library preparation that can be used for quantification and process control. So depending on the study setup, if you have try rate for your samples, we are enabling you to do an absolute quantification of your bacteria in your sample. We have been putting a lot of work onto target multiplex amplicon panels when we only want to look at a defined part of the community. And we're currently working very, very hard on metatranscriptomics, which is still a work in progress. And one of the development projects I'm personally most proud of that I've worked on since the center has been founded is host depletion. If you work in the metagenome field, you might be familiar with this. When you have a typical stool sample, most of the genetic material you will get out of an extraction is from bacteria. There's very little human shedding in fecal samples. So what you put into the metagenome pipeline is all you're interested in. So when we do the sequencing, we sequence all the genetic material in the sample. For fecal samples, this genetic material comes mostly from bacteria, but for blood samples and bodily fluids and swabs and biopsy tissues, we see very different abundances of biological material, of bacterial material compared to host DNA. So in a typical vaginal sample, which is a field we're focusing on, you would see 60 to 90% of host content. So in most studies, we are ethically not allowed to look at the host material. So 60 to 90% of the material we are sequencing, we are deleting immediately. So we are spending money. Still, I mean, sequencing got cheap, but not that cheap. Still 60 to 90% of the sample preparation costs go directly unlooked at in the bin, which has been very upsetting for me for the longest time. And this is not only for vaginal samples, this is still okay. If you look at biopsy samples, which we want to do more and more often in the gastroenterological field, you look at 99 to 99.5% host DNA. And there it gets just outrageous. There is like all of your sample preparation basically costs are basically for nothing. So we want to focus on the microbial communities and consequently reduce the costs for both for sequencing, data storage, and bioinformatics analysis. And early data of our host depletion assay shows that we are able to reduce up to 50% of the host treats, depending on the sample type. So this is also a bit of a messy plot, and but I will talk you a bit through it. So we are still having a bit of a hard time with biopsies, but for vaginal samples and saliva samples and spiked biopsies, we can see up to 50% removal of the host treats. And we think that host removal will be a breakthrough for boosting the sensitivity and to detect and characterize less abundant microbial components. And on the bottom of the slide, you see an overlay of detected species. And what you want to see when you do an assay like this, when you take out that much DNA, is you want to see that you're not actually removing DNA of interest. You don't want to remove bacterial DNA. And in the violet or rose colored circle, you see the core identified species. And then in the overlap in the blue circle, you see the species count after host removal. And you see that in all of the assays, we get a gain of knowledge. We need to do further investigation, because the second thing you don't want to do is you don't want to introduce species that haven't been there before. So in the final steps of our development, we're looking now into where those introduced species come from, if they come from us, or if they're actually a gain of knowledge. But up to this date, I'm very satisfied with the data. I'm very proud of the steps forward we have made, and I personally believe that we, in a bit more time, will revolutionize the field with opening up the possibility to deep sequence high host abundant samples as well. To get a bit back from the technological side and a bit into what you can actually do with all this technology, I'm going to talk a bit about what we do, what we're focusing on, what we're focusing our studies on. So in the studies of gastroenterology, we're mostly focusing on IBD, diverticulosis, polyps and tumors, and how they look microenvironmentally, and overall. So if there is a gastrointestinal microbiome composition that is normal, and how would that be different from patients that have one of those diagnoses? Is there a difference? Can we make out certain biomarkers that are different in those diagnosed states? What is the composition of the gut's microbiome in children with IBD compared to healthy children? Can we identify biomarkers that can improve the diagnosis of IBD, which up to date is still very diffuse and difficult? And is there can we change a dysbiotic gut microbiome profile to a healthy profile to cure IBD, for example? And one of those typical studies that we would use is a study that is called Oncobiome. So there we have 60,000 microbiome samples where we randomly send out fecal samples where we randomly send out fecal samples, or it's a random study where people submit fecal samples over the course of 30 years. I think 5 samples in total, if I'm not totally wrong. And then we do a register follow-up. So in Sweden, I'm not sure how this is in other parts of the world, but in Sweden we have those registers where all the samples are connected to a person, and every person has a unique personal number, and all samples are documented in different registries that we then as researchers can get access to. So we have been following the registry entries for the National Cancer Registry and the inpatient registers for the people that have submitted the samples over those years, and then cross-referenced individuals that got diagnosed with IBD or cancer that have deceased from one of those, as follow-up of one of those diseases, and then used the healthy patients or the patients without any entries in the register as control patients and sequenced them. And this is how you would compare or standardize a healthy community and compare it to different health outcomes with the registries to see if you can find biomarkers that would be indicative of a certain disease, or potentially be preferential for a health outcome. So the discovery of diagnosis and therapeutic tools in cancer and precision medicine is one of the main main aims of this project. So outcomes to find certain biomarkers that could be used as therapy optimization. So if certain biomarkers could have a positive or negative outcome on the toxicity or efficacy of certain therapeutic measures, biomarker discovery by the means of sequencing the stool samples. You could also use microbiome therapies, so fecal microbial transplantations, to change the community into a certain state that might be beneficial for a certain health state or health outcome. And then in the end, hope that with the means of computational biology, we could adapt those consortia that in the end are hopefully synthetic in a way that they would be adapted to a certain individual. Another one of our main research fields is women's health. And there we are looking into different questions, or a bit different field of questions. We have one study that is focusing on the question if the microbiome acquisition already starts in utero. We're also looking into the effects of the menstrual cycle and hormonal contraceptives to the microbiome. What composition of the microbiome is normal in healthy women of different ethnicities, if there is a microbiome profile that can be associated with pregnancy complications, and if we can identify patients that are at risk for preterm birth or pregnancy loss, and if there is a role of the microbiome in endometriosis and fertility. And our flagship project that I would like to talk you through is the Swedish Maternal Microbiome Project, and the overall aim is to assess the association between the maternal microbiome during pregnancy and postpartum, and the risk of maternal and neonatal adverse events. And the first main research question that we have been taking up in this under this umbrella is if there is a microbiome profile that can be associated with an increased risk for preterm birth and pregnancy loss. And the study was completely conducted from our center. So from the questionnaire design to the sampling material preparation, this is completely under our umbrella. And the samples we have been taken in the studies, or we have taken in the study, is fecal samples, vaginal samples, and saliva samples before week 20 of the pregnancy. Another set of samples between week 28 and 30, and then another set of samples 5 weeks postpartum, each with a questionnaire. And then postpartum, we also collected a fecal sample from the baby. So all in all, we have, in the study, we were able to recruit over 5,000 mothers and 3,000 children that were born between 2018 and 2021 in Sweden, which accumulates to a bit over 50,000 samples in this study. And while we were very fortunate and have a lot of very healthy pregnancies and healthy babies in the study, we also, due to the extensive amount of pregnancies that we have in the study, we also have significant amounts of pregnancy complications, both diagnosed and self-reported pre-existing diagnoses, that actually facilitate us to have significant numbers to pinpoint biomarkers that are helping, or that could help in the future, to determine by the means of a vaginal swab, determine risks for preterm birth or early term births or miscarriages. We also offer this service, as I mentioned right in the beginning, as a company today, as a state facility. We are only capable to work with collaborators within the field, and we decided to spin this out and take the 10 years of research and offer them to the field. So if you're interested in acquiring sequencing services from us and make use of our experience for your project, you're very welcome to contact me. And there I stop the own advertisement. And I start to thank my team that has made all of this work possible, that is very heavily involved in development of the pipeline and the processing of the samples, and has been just a wonderful support, and I would not know what I would do without them. But we are also more than LabCore. The LabCore unit is just part of the center, and the center is all of those lovely people that have given us a great environment to work in, and lots of support and help, and I'm happy to be part of this group. And with that, I thank them and thank you for listening, and give back to Kiara to answer your questions.
Thank you, Maike. As a reminder to webinar participants, if you have a question, please type it into the Q&A box. It's in the control panel. And would also like to ask attendees to take a moment after the webinar is ended to take our exit survey and give us your feedback. So we'll start the Q&A portion of the webinar now. And also joining us is Wenxi Lau, the Director of Product Marketing at Complete Genomics. And the first question is, I think, more for like clarification, just kind of like, why can't you sequence host DNA in conjunction with the target species DNA, or if it's, you know, a virus or RNA, or sorry, do I need to repeat? Yeah,
yes, please.
When you're talking about the reducing the host DNA, you said that you can't sequence it, that it's there's some like an ethical issue there, and I think someone was just a little confused as to why you couldn't do that.
Yes, you can sequence it. So sequencing is not a problem. You're just not allowed to look at it. So there are different ethical considerations looking at human data today. There are different ethical considerations looking at human data compared to looking at microbiome data. So when you start a study, not dependent on which study, you need to do an ethical application. At least that's how it's in Europe. I presume it's all over the world like this. And those ethical applications and considerations and how you handle the data is different for humans and bacteria. So when we do microbiome studies and make ethical applications for those studies, we normally do not apply to look at the human data just because it's a more tedious process, and we are not always interested in it. So if you do not apply to look at human data, then you're not allowed to. So you can sequence it, you're just not allowed to look at it.
Got it. And if you're not interested in it, why go through the process?
Yes. Also, it's a lot of data. That's I mean, it takes significantly more time, and processing time is money. And yes, so we want to save money and time.
What do you think is the next key step in improving automation?
The next key step in improving automation. I think we have come quite a long way. There is, without wanting to tell you too much, there is, I mean, robots will always need a bit of hand-holding, because even if you reduce the bias in handling the samples and you automate this part and you save your technicians your technicians' risks, then they still break on occasion and they need to be supervised. So there will always need and be a need for people in the lab. We have automated some of those processes as well. So there is AI involved in deciding if a plate is sitting right in a robot or not, and then alarming, but someone still needs to answer to this alarm. There is, however, if you listen very closely, I mentioned that the extraction is semi-automated. There is one part in the extraction process for metagenome sequencing that I have, as of today, not been able to automate. If I ever get the resources to do so, I would like to do it. It's a mechanical disruption of the bacterial cells that is still very difficult to automate. From a higher perspective, I think we have gone pretty far. The only thing that we are doing today by hand that we wouldn't need to, I think it's pretty fully automated. All the parts we do today in the lab manually, we do because we chose to, not because we have to.
Okay. And which reference databases do you use for bacterial identification?
I think for taxonomic classification, and I might have to make a guesstimate on this. I think it's Metaflan 5.
Okay.
I hope this answers your question. I'm not a bioinformatician. I try to keep as up to date as well on what they are doing as possible. So I think today they're using Metaflan 5, but I might need to get back to you on that.
Okay. We'll keep that person in mind then.
Yes. David, contact me if you want to talk more about this. Then I check more closely.
Gotcha. And can you elaborate a little bit more on how you do the environmental controls?
So, for example, we have studies where we compared cesarean births to vaginal births and inoculation with a smear sample for cesarean birth born children from the vaginal mucosa of the mother. And for there we take a plethora of samples, but they're very low microbial content samples because of the process. So we take swab samples of the rectal area of the mom. We take samples behind the ear of the child. around the aorta of the mom. We take saliva samples of a newborn are very difficult, so we take swab samples of the newborn, and there we do environmental controls to make sure that the process of the smearing, which microbial environment we have in the room that this is taking place, where the nurses have swabbed that is dipped in water, and they just walk with it through the room. I know it sounds a bit ridiculous, but that's the environmental control of the procedure. And then we have those controls also for our lab.
For a typical project, what's the turnaround time from receiving samples to getting results?
For a typical project, people ask this very often, and it depends very much. So when you do large metagenome studies, it is extremely important to have a very high throughput, because we are in a phase of technological development where methods change very rapidly, and the technology is changing very rapidly. A study design takes a very long time. So as you see, sometimes the sample, the recruitment takes 3-4 years. And when we get in all the samples, we want to process those 50,000 samples as fast as possible, because it's the process itself is including some sort of bias, how you extract the samples. There are a lot of publications that show that extraction lots have a huge impact, extraction kit lots have a huge impact on the microbial composition in the sample. So we want to use the same extraction kit lot for the whole study, and the same process in the lab with exactly the same kit lot numbers, have as little bias as possible. But a sample in, a sample out is a bit difficult, depends on the size of the study. So the throughput of the lab is approximately 1,500 samples a week. Okay. I would say that the lab process from getting it into the lab to getting it onto the server is taking approximately 3 days.
Okay. How do you decide, like when you're talking to someone about their project, how do you decide what samples to run on the DNB-seq G400 or on the T7 sequencer? What goes into that decision process?
Mostly the size of the study and the need for sequencing rates. So for if you want to sequence bacterial isolates, you need a lot less reads than you would need if you sequence a whole metagenome. If you only have bacterial isolates in order to fill a T7 flow cell with 6 billion reads, you would need very, very many. I've never done this, so I can't say it from the top of my head, but you wouldn't choose this platform. If you, it also depends on the funding of your study. So if you, so normally for fecal metagenome, we aim, depends a bit on the current trend, between 20 and 40 million reads per sample. But it depends on how many samples you need for your study and how big your funding is. So maybe you want to go for a bit of a shallower sequencing, and then you might want to choose the G400 and just press everything in there. It depends on how many indexes you need. Those are the considerations you need to make when you choose your sequencing platform.
Okay. Well, it looks like all the time we have for today. We'd like to thank our speaker, Maike Seifert, as well as Wenxi Liao, and our sponsor, Complete Genomics. If we didn't have time to get to your question, we'll try to follow up with our experts. And as a reminder, please look out for the survey after you log out to provide us with your feedback. And if you missed any part of this webinar or you want to listen to it again, an archived version will be emailed to all attendees. And thank you for joining us for this Genome webinar.